The Precambrian and the Cambrian period brought about dramatic environmental changes contributing to the emergence of multicellular life that saw behaviours such as extracellular feeding, locomotion, and predation benefiting from networks that facilitated information integration and communication. And the first manifestation of a nervous system was likely in the form of elementary nerve nets for information processing and the horizontal coordination of tissue movements. The earliest nerve nets would have been comprised of sensory and contractile cells that sped up and aided grazing on algal mats. Mechanosensory neurons, known as Kolmer-Agdor-like cells, are thought to have formed part of structures such as the mucociliary sole and the gastric pouch in protostome and deuterostome ancestors, and these later diversified into different specialized neuron types in the descendants of annelids and vertebrates. This is really important because the emergence of structures such as the mucociliary sole and the gastric pouch drove a switch to extracellular digestion, facilitating higher nutrient absorption, meaning more energy was then available for locomotion, which triggered the regionalization and the specialization of areas of the nerve net needed to control more targeted movements, as feeding requires substantial integration of ciliated, contractile, and secretory cells. Ultimately, this resulted in convergent centralization in ctenophores, cnidarians, and bilaterians. Nervous systems in both simple and complex form have evolved as a result of neurons acting as the primary source of transport for transmitters to relay information. Neurons themselves are derived originally from populations of neuroepithelial secretory cells that used chemical transmitters, which are the language of all nervous systems, for communication and the integration of behaviors in early animals without established synapses. The incredible spatiotemporal diversity and organization of neural networks and the huge variability of transmitter phenotypes indicates multiple genealogical origins. The convergent evolution of nerve nets and the diversity of forms of nervous system across metazoa shown in this diagram demonstrates the strong selective advantages provided by the rewiring and the repatterning of structures into integrative networks that allowed for the emergence of complex behaviors that came with drastic environmental changes. The independent evolution of neurons and centralized nervous systems arose in parallel with the rapid diversification of body plans as a result of recruitment and co-option of various transcription factors and gene regulatory modules found across many animal phyla. Despite sometimes in literature and by myself somewhat arbitrarily being labelled as simple and primitive, many phyla with nerve nets shown here in this diagram have a prominent concentration of neuronal elements and numerous and sometimes autonomous networks capable of orchestrating surprisingly complex and well-coordinated behaviours similar to those seen in higher organisms. Amphimedon is a primitive sponge that doesn't possess a nervous system. However, it's a brilliant organism for us to understand how the earliest neuronal networks may have evolved as a result of exaptation of components within the genetic toolkit. Amphimedon have crystalline and metabotropic glutamate receptors, which are two molecules that are found in the metazoan sensory system. They also possess voltage-gated ion channels, which are a superfamily of transmembrane proteins that are really crucial for executing roles such as the propagation of neuronal and muscular action potentials, promoting neurotransmitter secretion in synapses, and for allowing the permeation of sodium ions. These are thought to have evolved from calcium channels right at the base of the origin of the nervous system, and they are also found in many bacteria and archaebacteria. They have also been shown to express the Notch delta signaling system and a proneural basic helix loop helix gene in a manner that closely resembles the conserved molecular mechanisms of primary neurogenesis in bilaterians. They've also been found to possess vesicle trafficking modules and a relatively complete set of postsynaptic density homologs. These proteins are really important for processing neurotransmitters, which suggests that a complex protein scaffold possibly predates the earliest nervous systems. Overall, the presence of metazoan synaptic components, ion channels, neural genes, and developmental pathways in sponges and other aneural lineages for supporting sensory function suggests that the remarkable complexity and diversity of extant nervous systems arose from primitive, ancient organisms at the base of all animals.